Hello, hello, hello! Good day, Ma'am Grace and Psychmates! Kamusta kayo? Hope you're all good and ready to listen as the Group 2 welcomes you to Chapter 7, Mechanisms of Perception, Hearing, Touch, Smell, Taste, and Attention. The previous chapter introduced us the visual system. In this chapter, we will focus on the remaining 4 of the 5 exteroceptive sensory systems, namely the auditory, somatosensory, olfactory, and gustatory systems. Are you all ready? If ready na kayo, arat, let's begin. Hello again, Psychmates. I am Zaira, and my first topic is all about the principles of sensory system organization. Before we proceed to the 3 types of sensory cortex, let us first define what is sensory system. Sensory system is part of the nervous system responsible for processing sensory information. It is consist of sensory receptors, neural pathways, and parts of the brain involved in sensory perception. Sensory system include the following systems: visual, auditory, olfactory, somatosensory, and vestibular systems. Also, we will define what is sensory areas. Sensory areas receives information related to sensation, and it includes regions like primary, secondary, and association cortex. And here are the 3 types of sensory cortex. First is the primary sensory cortex. It is the area of sensory cortex that receives most of its inputs. The primary sensory areas receive information from peripheral receptors with only a few synapses interposed. The primary visual cortex is located at the caudal pole of the occipital lobe, mainly on its medial region, while the primary auditory cortex lies on the temporal lobe, where it includes a portion of the lower bank of the lateral sulcus. The primary somatic sensory cortex lies on the postcentral gyrus. On the other hand, the primary motor cortex lies within the precentral gyrus and contains neurons that project directly to the spinal cord. It mediates voluntary movements of the limbs and trunk because it contains neurons that projects directly to the spinal cord to activate motor neurons. The second type is the secondary sensory cortex. Comprises the area of the sensory cortex that receive most of their input from the primary sensory cortex of that system or from the areas of secondary sensory cortex of the same system. Surrounding the primary areas are the so-called higher-order secondary, tertiary sensory, and motor areas. These areas integrate information coming from the primary sensory areas. In contrast, higher-order motor areas send complex information required for a motor act to the primary motor cortex. The third type is the association cortex. Any area of cortex that receive input from more than one sensory system. Most input to areas of association cortex comes via areas of secondary cortex. Association cortex receive inputs from multiple areas. It integrate incoming sensory information and also form connection between sensory and motor areas. Because they are involved in organizing information that comes from various other areas of the brain, association areas are often linked to complex functions. These regions lies outside the primary, secondary, and tertiary. The association areas constitute, in primates, the largest area of cortex. Examples of association cortex. The parietal temporal occipital association cortex occupies the interface of these 3 lobes. It is concerned with higher perceptual functions related to somatic sensations, hearing, and vision. Information from these different sensory modalities is combined to form complex perceptions. Next is the prefrontal association cortex. occupies most of the rostral part of the frontal lobe. An important function of this area is the planning of voluntary movement. And last is the limbic association cortex. It is devoted mainly to motivation, emotion, and memory. It is located on the medial and inferior surface of the cerebral hemisphere in portions of the parietal, temporal, and frontal lobes. The interactions between these three sensory types, the cortex, as well as other sensory structures, are distinguished by three major principles: hierarchical organization, parallel processing, and functional segregation. We'll first talk about hierarchical organization. It is a system whose members can be assigned to specific levels or ranks in relation to one another. Religious groups are a prime example of a hierarchical system where one person sits at the top and the power is gradually shifted downwards. Sensory structures are organized in a hierarchy on the basis of the specificity and complexity of their function. As one moves through a sensory system, from receptors to thalamic nuclei, primary sensory cortex, secondary sensory cortex, and association cortex, one discovers neurons that respond optimally to stimuli of increasing specificity and complexity. Each level of sensory hierarchy receives most of its input from lower levels and adds another layer of analysis before passing it on top. Each level gets most of input from lower levels and adds further analysis to hierarchy. For example, receptors to thalamic nuclei to primary sensory cortex to secondary sensory cortex to association cortex, which makes up the hierarchy move up. Psychologists often divide the overall process of perceiving into two parts: sensation and perception. Sensation occurs when sensory receptors detect sensory stimuli. Perception, on the other hand, involves the organization, interpretation, and conscious experience of those sensations. Moving on to the second principle, functional segregation. It was once assumed that the sensory systems, the primary, secondary, and association areas, were all functionally homogeneous. In other words, it was assumed that all areas of cortex at any given level of a sensory hierarchy work closely to perform the same function. However, research has shown that the organization of sensory system is characterized by functional segregation rather than functional homogeneity. It is now clear that each of the three levels of the cerebral cortex in each system, the primary, secondary, and association, contains functionally distinct areas that specialize in different types of analysis. Alright, we are now down to the last principle, the parallel processing. In a serial system, information flows between components along a single path, much like a string through a strand of beads. However, evidence suggests that sensory systems are parallel systems with information flowing through the components via multiple pathways. Parallel systems include parallel processing, which is the simultaneous analysis of a signal in multiple ways by a neural network's multiple parallel pathways. The two essentially different types of parallel streams of analysis in our sensory systems are one capable of affecting our behavior without our conscious awareness and one capable of influencing our behavior with our consciousness. Here are the two models of sensory system organization. Sensory systems were ought to be hierarchical, functionally homogeneous, and serial in the 1960s. Subsequent research, however, has established that sensory systems are hierarchical, functionally segregated, and parallel. It is not shown in the current model, the model in the right side, the many descending pathways that are the means by which higher levels of sensory systems can influence sensory input. We can see that there is a big difference between the former model and the current model of the sensory system organization, but they are both in hierarchical organization. To end up my first topic, let's exercise our mind with a game called Fact or Bluff. I will give you 5 questions and you will just identify whether the statement is a fact or a bluff. I will give you 10 seconds to answer and you can put your answers in the chat box. Are you ready, psychmates? Umpisahan na natin. Number 1. Sensory system receives information related to sensation and include regions. Psychmates, it is a fact or a bluff? Yan, tama yan. Bluff, because the correct answer is sensory areas. Number 2. The 3 types of sensory cortex are primary sensory cortex, secondary sensory cortex, and association cortex. Psychmates, it is a fact or a bluff? Yan, tama yan. The correct answer is fact. Number 3. Most input to areas of association cortex comes via areas of secondary cortex. Psychmates, is it a fact or a bluff? Yan, tama yan. The correct answer is fact. Number 4. Sensory structures are organized in a hierarchy on the basis of specificity and complexity of their function. Psychmates, is it a fact or a bluff? Yan, tama yan. The correct answer is fact. Number 5. The current model of sensory system organization is hierarchical, functionally homogeneous, and serial. Psychmates, is it a fact or a bluff? Yan, tama yan. Bluff, because the correct answer is former model. And that brings us to the end of my first topic. Nag-enjoy ba kayo? Thank you so much, Psychmates, for your interest and attention. The next reporter is Miss Mejia.
The auditory system. The function of the auditory system is the perception of sound. Sounds are vibrations of air molecules that stimulates the auditory system. Humans hear only those molecular vibrations between about 20 to 20,000 hertz. So physical and perceptual dimension of sound. The amplitude, frequency, and complexity of molecular vibrations are most closely linked to perception of loudness, pitch, and timbre. Pure tones exist only in laboratories and sound recording studios. In real life, sound is always associated with complex patterns of vibrations. The figure illustrates that any complex sound wave can be broken down mathematically into a series of sine waves of various frequencies and amplitudes. As you can see, when added together, these sine waves produce this clarinet sound. So, the mathematical procedure for breaking down complex waves into their component sine waves are called Fourier analysis. So, for any pure tone, there is a close relationship between the frequency of the tone and its perceived pitch. However, the relationship between the frequencies that make up natural sound, which are always composed of mixture of frequencies, and their perceived pitch is complex. The pitch of sound is related to their fundamental frequency, or the highest frequency of which the various component frequencies of a sound are multiples. So remember, an extremely important characteristic of pitch perception is the fact that the pitch of a complex sound may not be directly related to the frequencies of any of the sound's components. Next topic is the ear. So when you say ear, you always remember hearing. So what is hearing? Hearing is dependent on a complicated set of procedures that convert sound waves in the air into electrical signals. These impulses are subsequently carried to the brain via our auditory nerve. So the illustration here shows how sound waves are processed throughout the ear. First, sound waves travel from the outer ear down to the auditory canal and cause the tympanic membrane, or also known the eardrum, to vibrate. Next, these vibrations are then transferred to the three ossicles, also known the malleus, incus, and the stapes. The vibration of the stapes trigger vibrations of the membrane called the oval window, which in turn transfers the vibration to the fluid of the snail-shaped cochlea. Then, the auditory receptors are mounted in the basilar membrane and the tectorial membrane rests on the hair cells. Accordingly, a deflection of the organ of Corti at any point along its length produces a shearing force on the hair cells at the same point. This force stimulates the hair cell, which in turn increases firing in the axon of the auditory nerve. Lastly, the auditory nerve carries the electrical signal to the brain, which turns it into a sound that we recognize and understand. Next is the auditory processing. So what is the auditory processing? Auditory processing is the ability to interpret the sound that one has heard. In central auditory processing, the brain must identify the incoming sound and make an analysis of those sounds and attach meaning to them. So, imagine yourself in a complex acoustic environment such as a party. The music is playing, people are dancing, eating and drinking, and numerous conversations are going on around you. Because of the component frequencies in each individual sound activate many sites along your basilar membrane, the number of sites simultaneously activated at any one time by the party noises is enormous. But, thankfully, your auditory system manages to sort these individual frequency messages into separate categories and combine them so that you hear each source of complex sounds independently. Next topic is the major pathways that lead from the ear to the primary auditory cortex. So there is no major auditory pathway to the cortex comparable to the visual system's retina geniculate straight pathway. Instead, there is a network of auditory pathways. The first relay of the primary auditory pathway occurs in the cochlear nuclei in the brainstem, which receive type 1 spiral ganglion axons, or also known auditory nerve. The second major relay in the brainstem is in the superior olivary complex. Then, leaving this relay, a third neuron carries the message up to the level of the inferior colliculus. These two relays play an essential role in the localization of sound. Then a last relay before the cortex occurs in the medial geniculate body. It's here that an important integration occurs in preparation of a motor response. And then the final neuron of the primary auditory pathway links the thalamus to the auditory cortex, where the message is already largely decoded during its passage through the previous neurons in the pathway. Next topic is the subcortical mechanisms of sound localization. So, the medial and lateral superior olives project to the superior colliculus as well as the inferior colliculus. In contrast to the general tonotopic organization of auditory system, the deep layers of the superior colliculi, which receive auditory input are laid out according to a map of auditory space. The superficial layers of the superior colliculi, which receive visual input, are organized written topically. Thus, it appears that the general function of the superior colliculi is locating sources of sensory input in space. So many researchers interested in sound localization have studied barn owls because these owls can locate sources of sound better than any other animals whose hearing has been tested. They are nocturnal hunters and must be able to locate field mice solely for the rustling sound of the mice make in the dark. Not surprisingly, the auditory neurons of the barn owl's superior colliculus region are very finely tuned. So here are the lateral superior olivary and the medial superior olivary nucleus. So the lateral superior olivary nucleus is concerned with detecting the direction from which the sound is coming, while the medial superior olivary nucleus has a specific mechanism for detecting the time lag between acoustical signals entering the two ears. Next topic is none other than the auditory cortex. So recent progress in the study of human auditory cortex has resulted from convergence of functional brain imaging studies in humans and invasive neural recording studies in monkeys. Still, primate auditory cortex is far from being well understood. In primates, the primary auditory cortex, which receives the majority of its input from the medial geniculate nucleus, is located in the temporal lobe, hidden from the view within the lateral fissure. So primates' primary auditory cortex comprises three adjacent areas. Together, these three areas are referred to as the core region. Surrounding this core region is a band or called the belt of areas of the secondary auditory cortex. Areas of secondary auditory cortex outside the belt are called parabelt areas. There is about 20 separate areas of auditory cortex in primates. So there are two important principles of organization of primary auditory cortex. First, like the primary visual cortex, the primary auditory cortex is organized in functional columns. Second, like the cochlea, the auditory cortex is organized tonotopically. What sound should be used to study the cortex? So many neurons in auditory cortex respond only weakly to simple stimuli such as pure tones, which have been widely employed in electrophysiological studies of auditory cortex. This practice is changing, however, partly in response to the discovery that many auditory cortex neurons in the belt and parabelt areas of monkeys respond robustly to monkey calls. It has become apparent that natural sounds in general are better at eliciting response from the neurons in mammalian auditory cortex. So, there are two streams of auditory complex. So, researchers have proposed that just as there are two main cortical streams of visual analysis, there are also two main cortical streams of auditory analysis. Then, auditory signals are ultimately conducted to two large areas of association cortex, the prefrontal cortex and the posterior parietal cortex. It has been hypothesized that the anterior auditory pathway is more involved in identifying sounds, whereas the posterior auditory pathway is more involved in locating sounds. Auditory visual interactions. Association cortex is usually defined as areas of cortex where such interactions or association takes place. Much of the research on sensory system interactions has focused on interactions between the auditory and visual systems, particularly on those that occur in posterior parietal cortex. Functional brain imaging is widely used to investigate sensory system interactions. One advantage of functional brain imaging is that it does not focus on any part of the brain. It records activity throughout the brain. So functional brain imaging studies have confirmed that sensory interactions do occur in association cortex, But more importantly, they have, they have repeatedly found evidence of sensory interactions that the lowest level of sensory cortex hierarchy in areas of primary sensory cortex. So where does the perception of pitch occur? This seemed like a simple question to answer because most areas of auditory cortex have a clear tonotopic organization. However, when experimenters used sound stimuli in which frequency and pitch were different, this information led Bender and Wang to probe primary and secondary areas of monkeys' auditory cortex with microelectrodes to assess the responses of the individual neurons to missing fundamental stimuli. They discovered one small area just anterior to primary auditory cortex that contained many neurons that responded to pitch rather than frequency, regardless of the quality of the sound. The same small area also contained neurons that responded to frequency, and Bender and Wang suggested that these areas was likely to place where frequencies of sound were converted to the perception of pitch. Auditory cortex damage. Most studies of the effects of auditory cortex lesions have assessed the effects of large lesions that involves the core region and most of the belt and parabelt areas. Given the large size of lesions in most studies, the lack of severe permanent deficits is surprising, suggesting that the subcortical circuits serve more complex and important auditory functions than once assumed. And although the effects of auditory cortex lesions depends somewhat on the species, the effects in human and monkeys appear to be quite similar. The major permanent effects are loss of the ability to localize sound and impairment of the ability to discriminate frequencies. The effects of a unilateral auditory cortex lesion suggest that the system is partially contralateral. Deafness is one of the most prevalent human disabilities, and according to the World Health Organization, an estimated 360 million people currently suffering from disabling hearing impairments, but total deafness is rare, occurring in only 1% of hearing impaired individuals. There are two common classes of hearing impairments. One is the damage to the ossicles, or also known as conductive deafness, and the other one is the damage to the cochlea, or the nerve deafness. Age-related hearing loss features a specific deficit in hearing high frequencies. That is why elderly people often have difficulty distinguishing S, F, and T sounds. They can hear people speaking to them, but often have difficulty understanding what people are saying. Hearing loss is sometimes associated with tinnitus or ringing of the ears. When only one ear is damaged, the ringing is perceived as coming from that ear. However, cutting the nerve from the ringing ear has no effect on the ringing. This suggests that changes to the central auditory system that were caused by the deafness are the cause of tinnitus. Some people with nerve deafness benefit from cochlear implants. Cochlear implants are bypass damage to the auditory hair cells by converting sounds picked up by the microphone on the patient's ear to electrical signals, which are then carried into the cochlea by the bundle of electrodes. And that's all for the auditory system. I am Alethea Mejia, and the next reporter will be Miss Lotero.
First, let's talk about the cutaneous receptors. These are a type of sensory receptors that are found in the skin. The first is the free nerve endings. It is the simplest cutaneous receptors. These are neurons ending with no specialized structures on them and are particularly sensitive to temperature change and pain. Next is the Pacinian corpuscles. These are the largest and deepest cutaneous receptors. They adapt rapidly and respond to sudden displacements of the skin, but not to constant pressure. Lastly, we have the Merkel's disc and the Ruffini endings. They both adapt slowly and respond to gradual skin indentation and skin stretch respectively. Somatosensory information ascends from each side of the body to the human cortex over several pathways, but there are two major ones: the dorsal column medial lemniscus system and the anterolateral system. The dorsal column medial lemniscus system tends to carry information about touch and proprioception, and the anterolateral system tends to carry information about pain and temperature. When Penfield mapped the relation between each site of stimulation and the part of the body in which the sensation was felt, he discovered that the human primary somatosensory cortex, or SI, is somatotopic, meaning it is organized according to a map of the body surface. The somatotopic map is commonly referred to as the somatosensory homunculus, or homunculus meaning little man. Somatosensory system and the association cortex. Somatosensory signals are ultimately conducted to the highest level of the sensory hierarchy to areas of association in prefrontal and posterior parietal cortex. Posterior parietal cortex contains bimodal neurons, meaning these are neurons that respond to activation of two different sensory systems, and these respond to both somatosensory and visual stimuli. Somatosensory agnosia. There are two major types of somatosensory agnosias. One is the astereognosia, which is the inability to recognize objects by touch, and the other is asomatognosia, which is the failure to recognize parts of one's own body. Asomatognosia is usually unilateral, affecting only the left side of the body, and it is usually associated with extensive damage to the right temporal and posterior parietal lobe. The rubber hand illusion. The rubber hand illusion is a feeling that an extraneous object is part of one's body. A volunteer's hand is hidden from view by a screen. The rubber hand is placed next to the hidden hand. In less than a minute, many volunteers begin to feel that the rubber hand is part of their body. The neural mechanism for this illusion is unknown. Functional imaging studies have suggested that the association cortex in the posterior parietal and frontal lobes play an important role in this induction. Perception of pain. A paradox is a logical contradiction. The perception of pain is paradoxical in three important subsections. Adaptiveness of pain. Pain is important in survival, and it is a response to potentially harmful stimulation of any type. Next, lack of clear cortical representation of pain. It has no obvious cortical representation. And lastly, descending pain control. Most compelling of all sensory experiences can be so effectively suppressed by cognitive and emotional factors. Neuropathic pain. What is neuropathic pain? It is a severe chronic pain in the absence of a recognizable pain stimulus. The typical case of neuropathic pain develops after an injury. Although the exact mechanisms of neuropathic pain are unknown, it is somehow caused by the pathological changes in the nervous system induced by the original injury.
Hello everyone, my name is Ruth Pascual and I'm gonna talk about the second to the last topic of mechanism of perception, which is the chemical senses, smell and taste. So, smell and taste are referred to as the chemical senses because they monitor the chemical content of the environment. Olfaction or smell is the olfactory system's reaction to airborne substances carried into the nasal passages by inhalation along receptors, while gustation or taste is the response of the gustatory system to chemicals in solution in the oral cavity. First off, before we dig deeper to the olfactory and gustatory system, what are the adaptive roles of the chemical senses? In humans, the main adaptive role of the chemical senses is the evaluation of potential foods, especially in natural environments. According to Yarmolinsky, Zucker, and Riba 2009, one role of the gustatory system in humans is to identify nutritionally important and potentially toxic substances in food, triggering actions that lead to acceptance or rejection of possible food sources. However, in other species, chemical senses play a major role in regulating social interactions. They release pheromones to influence the physiology and behavior of the other members of the same species. And these pheromones are detected by sensory neurons, specifically olfactory neurons, that are preset to promote an array of social and survival behaviors. Just for example, in the experiment of Murphy and Snyder, they show that the sexual and aggressive behavior of hamsters is under pheromonal control. And normally, male hamsters attack and kill unfamiliar males that are placed in their colonies, whereas they mount and impregnate unfamiliar sexually receptive females. However, male hamsters that are unable to smell the intruders engage in neither aggressive nor sexual behavior. So, in their experiment, they swabbed a male intruder with the vaginal secretions of a sexually receptive female before placing it in an unfamiliar colony. And in doing so, rather than kill and attack the unfamiliar male because they did not smell a male one, but a sexually receptive female, they converted it to hamster lust. Let's talk about olfactory system. Olfactory system is responsible for the sense of smell or olfaction. So basically, the inhaled airborne chemicals are converted into electrical signals, and the olfactory receptor cells, which are located in the olfactory mucosa, send them to the olfactory bulbs where they synapse on neurons that project via the olfactory tracts to the brain, where they are interpreted as odors. For decades, it was actually assumed that there were only a few types of olfactory receptors. But now it was discovered that in humans, there are about 300 different receptors, and this enables the olfactory system to recognize an enormous number of odorants. In mammals, there is only one type of receptor protein molecule that the olfactory receptor cell has, and are found in the membranes of the dendrites of the olfactory receptor cells, where they can be stimulated by the circulating airborne chemicals in the nasal passages. All types of receptor appear to be scattered throughout the mucosa, providing no clue about the organization of the system. So, the axons of the olfactory receptors terminate in discrete clusters of neurons that lie near the surface of the olfactory bulbs, and these clusters are called the olfactory glomeruli. And each glomerulus receives an input from several thousands of olfactory receptor cells, all with the same receptor protein. And because of the systematic topographic organization is apparent in other sensory systems, researchers have been trying to discover whether glomeruli sensitive to particular odors are arrayed systematically on the surfaces of the olfactory bulbs, and indeed, the evidence indicates that there are a systematic layout. The first evidence is there is mirror symmetry between the left and right olfactory bulbs glomeruli sensitive to particular odors tend to be located at the same sites of, on the two bulbs. The glomeruli sensitive to particular odors are arrayed in the olfactory bulbs in the same way in different members of the same species, just for example the mice. And the last evidence is the layout of the glomeruli is similar in related species, example, rats and mice. And although it is clear that the olfactory bulbs are organized topographically, the that topographic principle according to which the glomeruli are arrayed has yet to be discovered. And this and this topographic organization of the olfactory bulbs are called the chemotopic map. New olfactory receptor cells are created throughout each individual's life to replace those that have deteriorated. Each new olfactory receptor cell survives only a few weeks before being replaced. Each olfactory bulb projects axons to several structures of the medial temporal lobes, including the amygdala and the piriform cortex. The piriform cortex is considered to be primary olfactory cortex, but this designation is somewhat arbitrary. The olfactory system is the only sensory system whose major sensory pathway reaches the cerebral cortex without first passing through the thalamus. There are two major olfactory pathways that leave the amygdala piriform area. One projects diffusely to the limbic system. The limbic pathway is thought to mediate the emotional responses to odors. This is actually why, when we smell a familiar odor, we remember a certain memory of it. And the thalamic orbitofrontal pathway is thought to mediate the conscious perception of odors. And the other projects via the medial dorsal nuclei of the thalamus to the orbitofrontal cortex, which is the area of cortex on the inferior surface of the frontal lobes next to the orbits. The gustatory system is responsible for our sense of taste. The taste buds are where our taste receptor cells are found in the tongue and in parts of the oral cavity, and they typically occur in clusters of 50 to 100. On the tongue, taste buds are often located around small protuberances called papillae. The 50 to 100 receptor cells that compose each taste bud come in several types and subtypes, the functional significance of which is unknown. In each taste bud, only one of the receptor cells, the presynaptic cell, synapses onto the neuron carrying signals away from the bud. Communication among the other cells of a taste bud appears to occur via gap junctions. Like olfactory receptor cells, gustatory receptor cells survive only a few weeks before being replaced by new cells. So how is taste encoded by the taste buds? It was once assumed that there are only 5 different kinds of taste receptor cells, one for each primary taste, and that all taste would be encoded by the pattern of activity in the 5 types. The primary tastes are assumed to be sweet, sour, bitter, salty, and umami or savory, but a case can be made of others. One serious problem with this theory is that many tastes can be created by a combination of these 5 primaries. Major advance in the study of the taste transduction occurred with the discovery of various G protein-linked receptor proteins embedded in the membranes of taste receptor cells. One receptor protein responded to umami, 2 to sweet, and 30 to bitter. There does not appear to be a G protein-linked receptor protein for salty. Salts influence receptor cells by entering them through a specific type of sodium ion channel. The receptor mechanism for our sour is currently unclear. The mechanisms by which the responses of particular receptor proteins are translated into particular tastes are not well understood. Once many receptor proteins were identified, two important features of the gustatory system became apparent. First, there appears to be only one receptor protein per taste receptor cell, and second, taste receptor proteins are not restricted to the oral cavity. Many are found in the throat, esophagus, and lungs. There are three major pathways of the gustatory system, and gustatory afferent neurons leave the mouth and carry information as part of the facial nerve, which are found in front of the tongue, glossopharyngeal nerve, the back of the tongue, and the vagus, back of the oral cavity. All these fibers terminate in the solitary nucleus of the medulla, where they synapse on neurons that project to the ventral posterior nucleus of the thalamus. The gustatory axons of the ventral posterior nucleus project to the primary gustatory cortex, which is in the insula, an area of cortex hidden in the lateral fissure. A different area of primary gustatory cortex represents each taste. The secondary gustatory cortex is in orbitofrontal cortex, and unlike the projections of other sensory systems, the projections of the gustatory system are primarily ipsilateral. The archaic five primary component processing theory of taste implied that each gustatory receptor and neuron is broadly tuned, which responds to a wide range of taste. However, the rapidly accumulating list of receptor molecules and mechanisms suggests that each gustatory receptor is narrowly tuned, which responds to only one taste, or at least to a very few of them. Majority of receptor cells except the presynaptic cell have been found to be narrowly tuned, which is consistent with the fact that each cell has only one type of receptor protein molecule. An example of broad tuning is the presynaptic cell, which integrates signals from all of the receptor cells in its taste bud, and as a result, the majority of presynaptic cells are broadly tuned. Anosmia is the inability to smell, and its most common neurological cause is a blow to the head that causes a displacement of the brain within the skull and shears the olfactory nerves where they pass through the cribriform plate. Less complete deficits in olfaction have been linked to a wide variety of neurological disorders, including Alzheimer's disease, Down syndrome, epilepsy, multiple sclerosis, Korsakoff syndrome, and Parkinson's disease. Ageusia is the inability to taste, is actually rare, presumably because sensory signals from the mouth are carried via three separate pathways. However, partial ageusia, limited to the anterior two-thirds of the tongue on one side, is sometimes observed after damage to the ear on the same side of the body. This is because the branch of the facial nerve that carries gustatory information from the anterior two-thirds of the tongue passes through the middle ear.
Hi Psychmates, I'm back. My second topic is all about selective attention. Selective attention refers to the processes that allow an individual to select and focus on particular input for further processing, while simultaneously suppressing irrelevant or distracting information. Example, listening to your favorite podcast while driving to work. Selective attention has two characteristics. It improves the perception of the stimuli that are its focus, and the second one is it interferes with the perception of the stimuli that are not its focus. Here is an example from the book. If you focus your attention on a potentially important announcement in a noisy airport, your chances of understanding it increase, but your chances of understanding a simultaneous comment from a traveling companion decrease. Attention can be focused into two different ways: by internal cognitive process, endogenous attention, or by external events, exogenous attention. One another important characteristic of selective attention is cocktail party phenomenon. Cocktail party can refer to a similar phenomenon where highly pertinent stimuli, such as our name, can immediately grab our attention, despite not previously paying the conversation any attention at all. For example, while you are driving, you focus on all cars moving around you, the curvature of the road, and the distance from the traffic light. However, you turn a blind eye to an empty billboard that just passed. It was bigger and brighter than any other object in your focal point, but your brain ignored it. This phenomenon suggests that your brain can block all stimuli except those of a specific type from conscious awareness while still unconsciously monitoring the blackout stimuli in case something requires your attention. Have you ever been so engrossed in a conversation at a party that you fail to notice that someone new is standing only 6 inches away from you, attempting to catch your attention? You could have been so preoccupied that you didn't notice someone approaching you. However, it is also possible that you were experiencing change blindness. Change blindness is a condition in which a person fails to notice visual changes in their surroundings despite the fact that they are frequently quite obvious. In cases of change blindness, the person will likely miss large changes, such as someone standing next to them, waving their hand, rather than small or insignificant changes. Neural mechanism of attention. Prefrontal cortex and posterior parietal cortex play major roles in directing top-down attention. Moran and DeSimone 1985 first to demonstrate the effects of attention on neural activity. They trained monkeys to focus their attention on colors red and green and recorded the neural activity in a pre-striate area that was a part of the ventral stream and particularly sensitive to colors. In the experiment, when the monkey trained to focus their attention to red, the response to red was increased and to green decreased. The opposite happened when the monkeys attended to green. Corbetta and colleagues 1990 conducted an experiment to humans also focusing on attention using functional brain imaging techniques. Their participants to distinguish the collection of stimulants based on their movement, color, and shape. The areas of the ventral stream increased in activity in response to shape and colors, and attention to movement increased activity in an area of the dorsal stream. Their study of attention in human volunteers was conducted by Unger-Leder and Huxley 1994. They asked the volunteers if the faces that they showed belonged to the same person and if they are in the same position. The regions of the ventral stream were more active where they are attending to the identity, and the regions of dorsal stream were more active when the volunteers are attending to the position. Selective attention works by strengthening the neural responses to attended to aspects and by weakening the responses to others. This dual mechanism has been termed a push-pull mechanism. Some neural mechanisms of attention involve a surprising degree of neural plasticity. the receptive fields of visual neurons, which had been assumed to be a static property of visual neurons, can be shifted by spatial attention. Wommelsdorf and colleagues 2006 found that the receptive fields of many of the neurons shifted towards points in the visual field to which the subjects were attending. Similarly, Rose 2008 found that visual receptive fields of inferotemporal cortex neurons shrink to become little more than the size of objects on which they are focusing. Regardless of the particular stimuli that have been used to study the neural basis of attention, visual, auditory, gustatory, olfactory, and somesthetic, the mechanism have been the same. Attention is activated by circuits in prefrontal and parietal cortex that enhance activity in task-relevant sensory circuits and suppress activity in relevant sensory circuits. And last but not the least, simultanagnosia, the ability to perceive individual elements of a scene without being able to synthesize the overall meaning of the scene is described as simultanagnosia. A study found dorsal simultanagnosia, which is caused by bilateral parieto-occipital damage, including the intraparietal sulcus and bilateral visuospatial white matter pathways, and is best described as a restriction of visual attention in which the patient is only aware of one object at a time. Despite having normal visual acuity, the attentional deficit of simultanagnosia can be so debilitating that patients are frequently described as functionally blind. Patient may see an ant or an elephant, but only one object at a time, and are unable to localize or interact with them using visual information. Again, to end my topic, we will play again fact or bluff. Psych mates, you know the drill. Let's start. Number 1. Simultanagnosia refers to the process that allow an individual to select and focus on particular input for further processing. Psych mates, it is a fact or a bluff? Yan, tama yan. Bluff, because the correct answer is selective attention. Number 2. Exogenous attention is thought to be mediated by top-down neural mechanisms. Psych mates, it is a fact or a bluff? Yan, tama yan. Bluff, because the correct answer is endogenous. Number 3. Change blindness is a condition in which a person is unable to recognize subtle visual changes in their surroundings, despite the fact that these changes are typically quite visible. Psych mates, is it a fact or a bluff? Yan, tama yan. Is it a fact? Number 4. Simultanagnosia refers to the inability to recognize multiple elements in a visual presentation. Psych mates, is it a fact or a bluff? Yan, tama yan. The correct answer is fact. Number 5. Cocktail party is a phenomenon that refers to our ability to focus on one specific auditory stimuli while filtering out others. Psych mates, is it a fact or a bluff? Yan, tama yan. The correct answer is fact. Alright, that brings us to the end of Chapter 7, The Group 2 sincerely appreciate your attention today. Thank you again, site mates and Ma'am Grace, for your time. Have a good day, everyone!